Good morning, good evening, and good afternoon to everyone who's joining us today. We're really excited to talk about the role of AI in drug discovery and development. But before we get to that, we'd like to begin with a quick round of introductions. I'm Vandana, Research Director at TechVision, and I've had about 15 years of experience in research and consulting and have been associated with TechVision for about 8 years now. Really excited to talk more about AI in drug discovery and development later today. Would like our esteemed guest, Alex, to sort of introduce himself. We're really honored to have him here today. Over to you, Alex.
Hi, everybody. I would love to thank Frost & Sullivan for this opportunity to talk about AI-powered drug discovery. My name is Alex Javrankov. I am the founder and CEO of In Silico Medicine, a globally present clinical stage drug discovery company. We utilize generative AI to go from zero, from target discovery to generative chemistry to clinical trials, and we also do aging research. In addition to drug discovery, we're also working in petrochemistry, agrochemistry, and chemistry for sustainability, because at the end of the day, chemistry is chemistry. My background is in computer science. Computer scientist by training. Did my first two undergraduate degrees at Queen's University in Canada. I'm Canadian by citizenship, and then had an academic career in bioinformatics and biophysics. I started my career in GPU computing, so I do know a little bit about semiconductors. And then after I kind of quit semiconductors with a little bit of money, enough to last for a few years, I decided to spend the rest of my life on aging research, because that's the biggest problem that everybody has. All of you have it, and it will take everything away from us when we die. So also the major cause of all diseases. I try to contribute to aging research in many ways. One way is to discover dual-purpose therapeutics targeting aging and disease at the same time. So that's hopefully something that I will be able to touch upon in my presentation today.
Thank you, Alex. So we're really excited to hear more about it, especially about the aging research and your work there. We also are joined by Dr. Sudeep Basu, who's the Global Consulting Director for TechVision. Sudeep, would you like to say hello to everyone?
Yeah, thanks, Vandana. Alex, pleasure to have you here. Really an honor. As Vandana said, I'm Sudeep Basu, and I lead the technology advisory practice globally at Frost & Sullivan. Been with the firm for a decade and a half or more, or slightly over that. Joined right after my PhD as a rookie analyst in the Silicon Valley office. Heady days, those. The financial crisis had just started out, so really exciting times. But most exciting for me is this amalgamation of AI and drug discovery, and having a PhD in a previous life in computational drug discovery. This is really something I was looking forward to very eagerly. So look forward to your presentation and discussion post that. And over to Priyanka for her introductions. Priyanka.
Yeah, hi everyone. So I've studied biotechnology. I have a master's degree in biotech, and I'm doing this research from last 5 years. I have associated with Frost. It's going to be 3 years. So yeah, looking forward to an exciting session.
Thanks everyone for the introduction. Alex, we are really excited to learn more about what In Silico is doing. So we'd be happy to sort of look at the presentation and see what you have to talk about the role of AI in drug discovery and development. So if you could share your screen, it would be great.
Yeah, will do. Thank you so much for the opportunity to do so. I see some high-profile, familiar faces in the audience, so great to see you all. Thank you for joining. Bring your friends and share this presentation later on. I'll talk very briefly today, just for 15 minutes, on generative AI and automation for drug discovery. Tell you a little bit about the company and also about what excites me the most in the future. So first of all, our company is globally present, as I've explained. So we're headquartered in Boston, but we do have a site focusing on generative AI research in Montreal. I'm calling in from Abu Dhabi today, not very conventional place to set up a drug discovery company, but I encourage everybody to come and have a look. Wonderful country, and we have over 60 people here, scientists focusing on generative AI. We also have Taipei for quantum chemistry, quantum computing, Hong Kong for target discovery, and Shanghai and Suzhou for drug discovery, mostly focusing on small molecule design and preclinical and clinical experimentation. So it's a truly globally present operation. We have no nationalities, borders, etc. We serve the patients, and that's represented in our values. Patients first, relentless innovation, transparency and integrity, and highest quality and reproducibility. Please follow us on Google Scholar. If you follow me, you'll see at least one paper every week in mostly medicinal chemistry journals, but also high-profile biotechnology journals. We try to publish and make most of our research available. One greatest challenge that I see in drug discovery today, and many of you who are thinking about starting an AI drug discovery company, investing in one, or working with one, you will face this challenge 100%. And that is the difference in speed and pace at the AI side and drug discovery side. So here are some benchmarks that I have internally and also industry benchmarks on both sides. And when you are talking about AI and what I like to call software for science, the industry is moving extremely quickly. So if you develop a very capable model, if it's a foundational model, it's going to be current and capable for maybe 6 months. After that, it's going to be obsolete because nowadays foundational models are the fastest depreciating commodities on the market. So you invest a lot in model training, sometimes tens of millions of dollars, but it becomes very, very quickly obsolete because somebody else is going to tear you apart in benchmarks. So when we are looking at this problem from the kind of with the eyes of in silico, it takes us usually about 4 months to get to a prototype with a new chemistry or biology product. And time to product is sometimes over a year because we need to validate with real experiments. If you do it in a startup environment, gloves off and just code, you can develop a prototype in under 30 days. Some people develop a product. In biology and chemistry, it takes a little longer because, again, you need to validate. But now we are living in a world where AI can code and automation can help you validate. So we need to accelerate. And we are actually, so I'm trying to get to those kind of startup timelines in my software business. But in the drug discovery business, even if you are extremely fast, and I think, and again, it's my guess, super biased, I think that we've set some records and record benchmarks in AI-powered drug discovery. And our typical time to developmental candidate or preclinical candidate, that's pretty much everything up until IND enabling. And our PCC package includes 28-day non-GLP toxicity studies. So we've never failed in IND enabling to date, not a single time, because non-GLP toxicity studies include 2 species usually and are very comprehensive. So you cannot really go around 13 months significantly on average, on typical, because you need to do those toxicity studies to convince yourself and the regulators, but mostly yourself. And once you get to clinical stages, you move with the speed of traffic. You cannot really cut corners unless you're 100% sure that you are not going to give the patient the drug that is not effective. Usually, the safety is taken care of at the preclinical and phase 1 side. But you really want to ensure that there is efficacy. And the industry average to get to phase 2 with an absolutely novel target is 10 years. Like, look at GLP-1s, it's a 40-year journey to get to a drug. Phase 2 usually took them to 10 years. Small molecules, sometimes longer. And those timelines are not compatible with the pace of AI. What I'm trying to say is that if you are to discover a drug using the software that took you, 4 months to develop, this software is only going to be validated 5 years from now when you're done with phase 2. So the way we try to go around this is to have 2 separate businesses that target software and drugs at the same time. Because from one perspective, software without experimental validation is not software. You don't know whether it's working or not. You need experimental validation, but experimental validation is very long. So that is why we came up with this new model where we have the software business. We have software products. Think of it as AI that other people can use. And on their hands, it may work differently. They can perform different tasks. And we don't take their data. We just ask them to provide feedback, whether the tool worked or not, or it didn't. So if it didn't, usually a big dislike will indicate that it didn't work, and we would redo the model and improve it continuously for the entire community. So community level validation is extremely important because then you can see validation in many areas and also across different timelines. Once the models get validated, they get into model orchestra. We have now over 700 different models, predictive and generative. And those models can acquire MCP, so MPC model connectivity protocol, or you can develop your own APIs. MCP is just a new fancy word for an API. And via an API, those models could be orchestrated using large language models. So large language models are not great for specific drug discovery tasks yet, at least not all of them. You need to have domain-specific chemistry or biology models that go very deep and have significant validation in multiple experimental settings. But large language models can orchestrate. And we usually deploy agents that orchestrate different domain-specific models to perform different tasks. Also, we realize that many of those models can be used for agriculture, CO2 capture, new materials, new energy chemicals. And you can also utilize the knowledge and learnings from those sustainability initiatives to make your drug discovery better. So basically, this model allows us to equal the pace between software development, basically AI race, and drug discovery race. So you constantly get validation of your models. They become veteran models, and then they're being used by other models. This concept allowed us to create a very substantial suite of software covering biology, chemistry, medicine, and all things science. So you can go to insilico.com and look at what these things can do. Some are foundational models. I'm very excited about life models that understand life from cradle to the grave in multiple species, in mice, monkeys, and humans, trained on different types of biological data, something that ChatGPT would not understand well, but these things do, multimodal transformers called Precious 3 GPT. We also have very popular tool, generative chemistry, that allows you to generate molecules with these desired properties. And we have tools for all things science mostly focusing on CO2 capture and hydrogen storage. Using those tools that we usually publish in peer-reviewed journals and also make commercially and sometimes openly accessible, we developed a pipeline of therapeutic programs, because at the end of the day, the value of your company is in the molecules you create. Because the validation experiments cost you so much that it would not be commercially sustainable to just have the software business. You actually need to develop your own, discover your own drugs and then sell them. And they have to be of ultra high quality, because for big pharma and biotech that buy those molecules, they usually have higher standards for external in licensing than for internal R&D. So you need to ensure that the data package is superior than of a traditional biotech. And you also need to use AI to create the data package, to curate it, and of course, use super expert humans. So far, we've managed to demonstrate that a novel target that has never been in a clinic before, called TNIC, could be purposed towards idiopathic pulmonary fibrosis. We think that it's very implicated in aging as well. We generated a novel molecule for it. We didn't license or pick something from a library. It's true generation. And then we took it all the way to phase 2 complete with dose-dependent efficacy. And some of the drugs that we've developed, one at phase 1 was sold to Exelixis in a very substantial deal, $80 million cash upfront, USP1 inhibitor, and several others were licensed out as well. CAT6 and KIF18A and KIF-STL we partnered. Many of those drugs here are available for out licensing and partnering. So give me a call if you're interested in any one of them, but we are, of course, progressing them ourselves. This is kind of a more comprehensive pipeline. We also started adding some new, so as you get more and more experience and you understand that the models that you developed really work in experimental setting and the molecules are pristine, better than what you can develop utilizing just human intelligence. We started going into more venturous disease areas like pain. So now I have 4 programs in pain. Some molecules work better than morphine in experimental settings, in experimental models. And we started going after more popular diseases like obesity and metabolic diseases. There we did not reinvent the wheel. Sometimes you have to go after known targets. So one of our targets is, well, GLP-1. But the molecule that we are developing has the half-life of 1 week, which means that you can actually go around with once a week dosing if we are successful, which makes it super convenient, and it's oral small molecule. Let's see if it works. But this is, I guess, the most important slide during today's presentation, because it's about what AI can give you today in terms of drug discovery and development. So far, since we got our first significant funding round where we could actually try our own drug discovery and development, we've nominated 22 preclinical candidates, 10 reached clinical stage, 2 phase 2s, 1 completed phase 2A. Our typical time to developmental candidate is under 13 months. Our record was 9 months. That was absolute record. We did we selected the new target KIF-STL and purposed it towards immuno-oncology, discovered the novel molecule, all in the matter. So we started counting days, and then we had to complete the 28-day non-GLP tox and then go IND enabling. But before IND enabling, 9 months. That's a really, really fast timeline. And in 2022 alone, we nominated 9 developmental candidates. Usually big pharma in small molecule space, they can nominate just as much with much bigger budget. And actually since 2022, we slowed down in terms of nomination of developmental candidates, because you actually have to take those molecules in the clinical trials in order to demonstrate to pharma that they should buy them. So that is a very important benchmark. So this is the benchmark. So basically, if you are trying to use another methodology, you need to try to demonstrate that you can beat those benchmarks. If you cannot beat those benchmarks in several attempts, well, you can buy our software. It's commercially available. And I'll just show you very briefly now several good experimental case studies for novel drugs discovered using generative AI. So in 2018, we conducted a few experiments published in 2019. And this paper was, generative before generative became popular. It was on generative tensorial reinforcement learning. Yes, my highest cited paper to date. We did it with Alana Spurogudzik, who used to be our fierce competitor when he was at Harvard, but then now he is one of my closest friends at the University of Toronto. And we published this paper. So he was the last author. I was the first author. It became very popular. And in this paper, we had one of our partners, a CRO, nominate pull out of the hat a target with specific criteria, DDR1 kinase. In 21 days, we used generative tensorial reinforcement learning, synthesized 6 molecules only. And 46 days, we've completed the in vitro validation, metabolic microsomal stability assays, and went into mice for PK, all that in 46 days. So there, we, of course, got some criticism because the molecule was not super novel, the target was not super novel, but it was the proof of principle for generative tensorial reinforcement learning. So think about it as like ChatGPT 0, and we just showed that it can talk. Most people didn't realize what we did at that time, but the paper got pretty popular. And now you can see in a kind of third-party paper by Tom Blundell's, they looked at all of the models out there ever published and validated with input, output, task, target, hit rate, the result, right, nanomolar inhibitor, and the year published. So you can see that many of our models have been validated experimentally. So when you're talking about generative reinforcement learning, I actually highly recommend going to this paper. They also have the GitHub repositories where they constantly update this. And you can see the validation, because unless the model is validated, you shouldn't really trust it, even for our internal programs. We want to see several experimental validations before we pull the trigger on something that is going to cost us, $200 million. But you can also generate compounds without crystal structure. So if you want to see an example, I think that probably that was the first time anybody has used AlphaFold to go after a dark target without a crystal structure that was not previously used in a disease. So we used chemistry 42 to very rapidly, in 2 cycles of synthesis, generate several molecules for CDK20. The most potent one was 180 nanomolar. We tested it in vitro assays. This experiment took 50 days. So less than 2 months, which is pretty cool. And a pretty highly cited paper. Please have a look. We can also utilize AI to repurpose existing drugs. So one of the case studies I can show was a collaboration between Mary Chutkovitz, Jeffrey Rothstein, Lubai, and us, and also Professor Janke from Mayo Clinic at that time. So we accessed their curated dataset called AnswerLS, mixed it with primary LS patient brain samples. They have mostly iPSC derived data from patient-derived cells. Combined it, gave it to AI called PandaOmics. It's more than 20 different models that select targets. Came up with new targets, drugs to repurpose, and tested it in a fly model. Out of the 26 predicted targets here, and we can find the paper we very quickly published in Frontiers in Aging Neuroscience, wanted a quick publication. Out of the 26 predicted, 18 worked very strongly, so showed strong or moderate rescue of the ALS phenotype in fly eyes using RNAi experiments, and that was very encouraging. So then Lubai, a very famous Chinese American scientist, founder of 4B Technologies, used one of the targets and one of the repurposing candidates to enroll 64 patients in a kind of phase 2 equivalent study, principal investigator initiated study, 64 patients And now we're waiting for the data. So hopefully the manuscript is going to be soon in review. Prepare for a surprise and look at 4B technologies. But the most important validation to date, I can show where we can show that we can discover a novel molecule, novel target using generative AI and automation is this. And before I do this, I want to show you my lab. This is the real footage, not animation. You kind of walk in, it looks like a spaceship. It recognizes your face, tracks you, dimmed windows. I can undim them, see the robots. And we decided to connect AI with automation, where the robot can pick up the samples, process it, give it to small autonomous guided vehicles and even the humanoid. Put it in the incubator. Part of the sample can be processed in the imaging station, 4 or 5 types of imaging, and then 3 types of NGS, methylation, transcriptomics, and whole genome for every sample. We predict the targets utilizing the original data that we get from each sample based on the novelty criteria and also commercial tractability. Then we have we pick up the tool compounds for those targets that may have tool compounds and validate it experimentally. So basically incubate the original sample with those predicted compounds. And if AI did well, it gets rewarded. If it gets it poorly, it gets punished. And if we find some really promising targets that don't have a compound, but many of the compounds scored well, we prosecute it ourselves. And of course, now we're continuously improving this automated facility for a more rapid validation. Now I have our first humanoids being tested. So it's going to be a lot of fun for this year. And this automation piece was actually utilized in one of our papers that we recently published, a very conservative name, a small molecule TNIK inhibitor, targets fibrosis in preclinical and clinical models. And in this paper, we show how we utilized target selection AI, also prioritized the target for aging. So I wanted to make sure that we are hitting all our main aging pathways, utilizing a master regulator, generated a small molecule for this target, and also wanted to make sure that if there are any off-targets effect, they're also hitting aging, synthesized just 79 molecules, and went all the way to phase 2A, completing many, many different experiments. So everything up until phase 1 complete is in the paper. So now we have a paper coming up soon explaining the phase 2 results. And in the phase 2 results, we've demonstrated force vial capacity increases in IPF patients. So stay tuned, it's going to be good. And we're it's not typical for a biotech company to sit on the data when the data is good. So some people naturally assume that it's not great. But we decided to wait until a publication in a top peer-reviewed journal. So hopefully soon you're going to see it. And I'll skip a few slides, but if you are interested in the aging component of the study and also automation, we recently published a paper on aging and disease showing that this compound that we've identified and that completed the phase 2A study is also a xenomorphic agent, meaning it reverses the aging signatures in multiple cell types. And we have one other case study very similar where we went from the target to phase 1, published in Nature Biotechnology last December. Here we use the PhD inhibitor. So I'm just going to skip it for the matter of time, but it's again from 0 to phase 1. And in the paper, we have everything up until the start of phase 1. Now 2 phase 1s are complete. We have excellent safety. So now we're planning to do the phase 2 efficacy study. But what you need to know is generative AI is not magic. At the end of the day, all of us are not just TechBio companies. We're biotechs. We live and die by clinical trials. And if you develop a really good algorithm that you believe in, that you think is going to work, the best way to validate is to commit yourself for the next 10 years and try to make a drug, because that's what we are judged by, right? So you cannot just have fancy AI papers. I thought that you need those papers, and for the first 5 years of our existence, that's what we've been doing. But without the real drugs, without real validation, this effort is not going to go anywhere great. Finally, I want to invite you to our annual conference. Every year we organize an event in Copenhagen, 5 days, called ARD, where we talk about aging research and its role in drug discovery. It's about 180 speakers, so you're going to be in good company.
Great. Thank you for that wonderful presentation, Alex. Needless to say, our minds are blown in terms of the potential that AI has in unlocking drug discovery and development. If I could move on to the presentation today, we will be, please let me know when you can see my screen. Is it visible?
Yeah, we can see it, Vandana. Yeah.
Perfect. Great. So we'll be talking largely about intelligent drug discovery, and like what Alex already said, what AI helps do in drug discovery and development is reduce cost, improve data integration, improve the success rate, and accelerate time to clinic. It cannot definitely replace clinical studies at any point, and we would need that eventually for validation and new drug development, but it can definitely improve productivity and de-risk clinical trials. And that's what's happening from a TechBio perspective. We are talking about companies like IBM, which are largely tech focused. They're using Gen AI design for developing new antibody based drugs and are collaborating with large pharma like Boehringer Ingelheim to take this forward. And we see a lot of that happening in today's sort of timeline, and that is definitely going to increase going forward. The partnership between pure tech companies and large biotechs to accelerate drug development, and that is a trend that we're definitely going to see in the next 3 to 4 years. Sudeep, would you like to add any thoughts on it so far?
First of all, I think really fascinating presentation there from Alex, and then follow on, Vandana, what you mentioned in terms of testing and animal testing sort of being there. I do want to call out attention to a recent sort of notification from the FDA, which actually talked about the reduction of some of those testing requirements going forward and refinements or potential replacements to those approaches, including AI-based computational models of toxicity in cell lines and organoid toxicity testing in lab settings, which is called the new approach methods or NAMS data, right? And I think this is something we have to watch very closely, and is probably music to Alex's ears, given everything that he's described in his end-to-end workflows and the kind of astonishing efficiencies he's been able to generate there. But it gives you a sense of things to come.
Correct. I couldn't agree more. And with that, let's move on to the first poll question for today. So I'll read out the results for everybody who cannot see it on the screen. So the question that we had asked was, how can AI accelerate drug discovery and development? 44% of the users believe that it can accelerate drug discovery through faster drug screening. About 22% believe that it helps in de-risking clinical trial outcomes and accelerate drug launch. 44% believe that it impacts all the factors like faster drug screening, de-risking of clinical trial outcomes, accelerating drug repurposing, as well as accelerating drug launch. And about 33% believe that it helps in accelerating drug repurposing. So I'd say it's a fairly even split across all the answers. What do you think, Alex? Would you agree?
So I think that it's all of them, right? So actually, it doesn't matter, again, how you do it, right? You can use a Scotch tape. Nowadays, you would use some AI component in the process. So that's actually how many pharmaceutical companies avoid the question whether Like if you ask them, well, do you have an example where you used AI to go from zero to phase one in a certain amount of time? They usually would say, oh, no, we don't, because we use AI at every stage a little bit, right? But I think that AI can actually help you at all of those stages, but most of the time it helps you in early preclinical. That's where you can save additional 2 years and increase the probability of success.
Right, that's correct. Yeah. What do you think, Sudeep? Would you agree with that?
Yeah, no, I completely agree with that. I think it's all of the above. It's not a surprise to see that faster drug screening was picked up as one of the higher numbers, because that's where most people have been focused, and most of the effort in the past has been on that. But I think going forward, we'll see some of those stats shift out. And I would have seen more on accelerate drug launch, because essentially that's the ultimate goal. And if you can shorten the time and if you can shorten the dollar spend on that launch, and that is, I think, where the productivity gains from AI will really make an impact.
Right. It'll be really interesting to see how this evolves over time. If we were to do this webinar again, we'd definitely see a different set of answers, hopefully, in a few years from now. If we can sort of go on to the next slide somehow. Yeah, there it is. So Priyanka, would you like to sort of walk us through some of the key points here as with respect
Uh,
to the vertical integration of AI in pharma? Yeah.
Yeah, so I really feel AI is transforming the whole industry, pharma and healthcare as a whole, as it is accelerating several processes ranging from drug discovery and development to drug design, clinical trials, CMC and formulation. If we talk about drug discovery and development, I feel it is one of the key hotspot where so many companies are actively looking at integrating AI in the process. AI really help here in identifying novel targets, as we already discussed before. It helps in designing better drugs. It also help in this predicting drug behavior. So companies are leveraging this ML and generative based AI tools to really accelerate the drug discovery. Key companies in silico is of course and Recursion. Both the companies are leveraging AI for accelerating the drug discovery timelines. And apart from drug discovery, repurposing is also something where AI is really being useful. And both the companies are also exploring AI for drug repurposing. When we talk about precision medicine, AI integration is making huge progress here. The ability of AI to really analyze novel biomarkers and genetic data, patient-specific inputs, this really aids in personalized treatments. So the companies like Lenton Pharma and Concert AI is making successful discovery in the field of oncology. Clinical trial is something that is really getting a boost and so many benefits by integrating AI. In clinical trial, AI really had to optimize the clinical trial designs, outcomes. It help in uh, recruiting better patients, thus improving the overall outcomes. The companies like Sama AI and AI Cure are developing tools that can really accelerate the clinical trial timelines and help in the faster therapeutic launches. If we talk about imaging and drug design, you already mentioned IBM. So apart from IBM, Google Health is also here, and Google is leveraging AI for breast cancer screening. And apart from oncology, I feel the cardiovascular disease monitoring, heart health, this ECG and EEG, those technology are also involving AI. And the integration of AI in medical imaging is really transforming the diagnostics overall. And in CMC, its formulation is also something that is observing using AI being used here. So the Merck and the companies like this Persist AI. So Persist AI has developed this AI-enabled cloud lab that can really develop the formulation for a sustained release formulation in just 14 days, the process which usually takes months. So you can see how AI is transforming the drug formulation. And I feel drug discovery and development as all It's being just explored for small molecule for now, but I feel there is a scope and opportunities for the company to explore AI for biologics drug discovery as well. I would like to know Alex thought on this because yeah, we already discussed this before when I had initial call with Alex. So biologics has certain complexity when it comes to integrate AI with biologic discovery, but company like Erexion is one of the key player. It has most advanced candidate it is has candidate in phase 2, which is a peptide-based vaccine for melanoma, and company has active pipeline for infectious diseases also, including both bacterial and viral infectious diseases. So it could be if the drug launch successfully, it could be a great breakthrough, like biologic discovery leveraging AI. So yeah, the future is
Right.
looking bright. Yeah.
Yeah, yeah. Alex, what are your thoughts on the use of AI for biologics? It's definitely more complicated than it is for small molecules. Would you agree?
Oh, I would disagree with that.
Really? Oh,
Because for biologics, we actually have a software package called generative biologics that people use for biologics design. So we have not pulled the trigger on biologics ourselves. We do have some programs very early and collaborations in biologics. So biologics are language. Um, it is basically the string of amino acids, and before that, nucleic acids. Large language models are great at working with language, and we've already seen that. And then those large language models are paired with models or developed together with models that understand structure and many other things, so multimodal, they become super intelligent. And it's actually easier to design a piece of the antibody that will become a better binder to its target. And people have done that before many times over. There are approved biologics that were kind of designed using traditional machine learning. It's fact. You can replace a few amino acids and improve the properties of a drug, of your biologic. Um, Nowadays, if you look at the major progress, at least from the headlines perspective, where AI did extremely well, there were Nobel Prizes for that, is protein folding. And
right.
protein folding is basically biologics. So we've seen quite a bit of progress in this area. And right now, every pharma company that is designing biologics, they are in one way or another using AI. Many pharma companies decided to go biologics nowadays because of the Inflation Reduction Act, right? It's just a more protective strategy. But now, maybe with the new administration, it's not going to be as trivial. So small molecules may get, better treatment. Because at the end of the day, small molecules are much cheaper to make and much easier to transport and deliver to everybody around the world. For small molecules, there are so many more parameters, especially dealing with safety, that you need to account for, that you don't need to think about when you're discovering and developing biologics, right? So safety, small molecules may have secondary metabolites. They may have significant off-target effects it's very difficult to predict. Half-life, metabolic stability, many, many, many other things. So blood-brain barrier. With biologics, many of those problems are not problems, right? Because you've got very high selectivity. You just bind to the epitope you've designed the antibody to bind to. You know that you most of the time won't get into the brain. So you will have to get specific delivery mechanisms for that. And you actually don't need to think about so many parameters.
Right. That's a brand new perspective right there. So thanks for that, Alex. Sudeep, what are your thoughts on it?
No, I think
Biologics
really,
versus
really,
small molecules.
yeah, fascinating to hear that. I definitely think that the complexity of biologics is something that, obviously Alex alluded to. Also, the changes that are afoot right now with regards to various different policies. might impact that. I think, to sort of get into our Q&A segment, I'd really like to say that, Alex, you spoke about no national borders and boundaries for innovation. I think that message resonates strongly today for global collaborative science. Also, your journey from semiconductors to biophysics is fascinating. And like you said, hey, this is not magic. But as Arthur C. Clarke famously said, any sufficiently advanced technology is indistinguishable from magic. So you're close there, really close. It gives us great hope to see a significant portion of your therapeutic pipeline being solid tumors. I think in oncology, that's a really difficult space. And having collaborated with several startups in that area, we know that it is a very active area. And on the physician side, too, it's an area where they're looking for tools to fight this. And the level of automation you have famously demonstrated in your Suzo lab, Alex, I reckon, we could have partied harder during our PhDs. And your focus on robotics and AI is imminent. So sketch us a view, if you may, of the lab of the future, say, a 5 to 10 year horizon. What functions do you think, can be further automated? And where do you think human involvement might still be desirable in that time horizon?
So that's the question to me?
Yes, Alex, to you.
Yeah.
All right. So I think that most of the drug discovery and development process at some point in time will be automated. The question is about the time horizon. I think for the next foreseeable future, maybe next 10 years, we are likely to follow the very traditional paradigm that we've seen before. So even with the new efforts by the FDA announcing the kind of the new favorable policies toward early preclinical testing versus work on animals for going into human clinical trials, even that policy, if you think about it broadly, the FDA is one region, right? It covers the US. And of course, the world follows. But if you are developing a global drug and you want to conduct a global clinical trial, many other countries would actually want you to create a preclinical package that will have animal experiments. Also, if you are going to invest $200 million into a drug, not a small sum, you probably would want to make sure that you have substantial number of animal experiments to believe that the drug is likely to be successful and it's going to be safe. So I think that the traditional paradigm is not going to be broken anytime soon. The only way to improve it is to set the benchmarks. And that's what we are trying to do. So basically, commit to a drug discovery program, right? So by the way, when you do that, as a manager, just looking at myself, you need to ensure that you are ready to go for 5 to 10 years with one company, with one drug, ignoring a lot of nice shiny things like starting a foundational model company or doing a robotics company. When you are going to commit to something like this, you will need to commit several years of your life, many years of your life. And just you have to do it, and you need to show how long it's going to take you to get from zero to a specific stage. So the first kind of date with a molecule is the developmental candidate, right? If you're talking about grand scheme of things. Of course, if like small micro level, first date with a molecule will be synthesized and get the binding affinity. But on the grand scheme of things, you need to show that you can get to developmental candidate. And you need to demonstrate that many times over with different target classes, different molecules, build up the stats and say, okay, with AI and with automation now from 0 to 1, to the first date, I can go go in 13 months. This is how much it costs me, and this is the downstream probability of success, right? So 0 failures in IND enabling. This is my phase 1, phase 2, and hopefully phase 3 and probably approval. So once you get the first approval and get the money for where your mouth was all this time, and get enough budget to go bigger, you will go bigger. But the first time you do it, you need to do it extremely well, and preferably several times. Once you have built the stats, those benchmarks, you can continuously improve them. And if some of these steps become obsolete, so you don't need animal testing, great. You save additional 9 to 12 months, right? IND enabling studies plus maybe part of non-GLP talks. Great, but human clinical trials will probably still be required.
And Alex, thanks for sharing that very balanced and grounded view from somebody who's been a strong proponent of AI in drug discovery. It's refreshing to see that. And there's a question here from the chat, which I do want to address and honor their time and participation. So this question came in via chat. So in your experience, Alex, what distinguishes a platform that merely predicts targets from one that delivers actionable prescriptive insights? And how do you quantify that value?
Again, experimental validation. So you need to demonstrate that you can pick a target that is of moderate or high novelty and take it to clinical trials. Even before you go to clinical trials, you would need to demonstrate at least 2 animal models and multiple in vitro models that your target discovery platform worked. But you will not know until phase 2. So you need to demonstrate that it shows promising results and preferably very clear results in phase 2. Before that, your platform doesn't work. And when you have proven that it works in phase 2, it means that it works in that specific disease, in that specific area. So preferably you need to do several phase 2s.
All right, and one more question that came in from LinkedIn. This is, what's the most frustrating part of your current drug discovery's workflows and an area that needs improvement?
Well, several areas, but most difficult one is fundraising. So for this experimental validation, you really need to raise a lot of money. And that's the most difficult part because the investors kind of are split into several categories, and not everybody is using Frost & Sullivan. So use Frost & Sullivan. Many of them, they want to have the returns very quickly. And they like to think that the AI platform can deliver those returns. And they always get burned, right? Because the total available market for AI software in drug discovery is actually tiny. So it's tiny. It's not like, I don't know, depending on how you count, but probably less than $1 billion, significantly. So then comes the drugs, right? And tech investors don't understand that area. Biotech investors, they like to be very cautious. They're very, very cautious. So if you have any slight trace of novelty, you'll be more scrutinized, right? So you need to find that ultimate balance between novelty, confidence, commercial tractability, investability, plan Bs, right? So if you don't work in the primary disease, you can, put it somewhere else. So you're not a binary company. So fundraising is difficult. And this kind of, again, when you have to operate on a scale of a decade, not just, a few months or a couple of years, what tech investors are got used to, it's a completely different game. So very difficult to fundraise. And we were lucky enough to kind of break through this ice. And now we've not monopolized, but I guess we've taken the lead in this industry. So people are using our benchmarks when evaluating companies, and hopefully they kind of understand the business a little bit better. Now it's a little bit more mature, so it's a little bit easier to fundraise, or harder for people who are basically doing outright fraud, all right, or telling the stories that here is my small folding model or large folding model. I'm going to change drug discovery and cure all diseases. Much more difficult to fundraise for them. Well, some it's easier. Second biggest challenge nowadays is geopolitics. So the world has gone crazy recently. You've seen what is happening, right? And I'm not a silent passenger. I like to talk about that. Life is the fundamental human right. Without life, there is no more rights to be desired. And the biomedical infrastructure is distributed around the world. You can design, so I can invent a drug in seconds. Right now in front of you, I can come up with a novel target, generate a molecule, but then I need to synthesize, I need to test it, I need to find the talent with specific animal models that will confirm the hypothesis. I need to talk to the key opinion leaders. I need to perform animal trials, monkey studies. I need to go and do a human POC, preferably quickly. So this new trend towards deglobalization is hurting the industry badly. And it's not about AI. AI, we can do it in one place anywhere. But in terms of the infrastructure, this decoupling is dramatic. And we need to ensure that we're true to our values. It's very easy to get distracted by ideology. But it's extremely important to know that we are responsible for patients, and that's why we shouldn't think about disconnecting. We should think about how to collaborate, at least in this particular place.
Thank you. Thank you for your message, Alex, and in keeping science and innovation forefront and collaborative science around the world being sort of the need of the hour, and thinking along those lines. I think we're having several more questions to go through, but we're unfortunately out of time. What a fascinating discussion this has been. Handed back to our coordinator.
And just to quickly summarize what we do within TechVision, I'd like to talk about our transformational growth journey. What we look at is the mega trends that influence tech developments across several technology clusters, and we see how that transforms industries globally. Every year, we have this annual exercise wherein we identify top 50 global technologies across 9 different technology clusters using a combination of qualitative as well as quantitative parameters. We assess over 72,000 technologies. In a year, we look at over 3,000 entities and put in several thousand hours of research efforts and try and assess each technology based on their IP activity, funding, and several other factors to come up with a list of 50 technologies that will have a great impact in the near term within 3 to 4 years from now. And we also pick 2 technologies that are in early development stage across each cluster. So a total of 18 futuristic technologies that will have an impact 7 to 8 years from now. If you want to learn more about it, do reach out to us. What we've been able to do successfully for 15 years within TechVision is identify technologies much before they become truly disruptive. One such example is that we were able to identify mRNA drugs in Jan 2019 as a part of our top 50 exercise 2 years before we were affected by the COVID-19 pandemic. We've been able to do that for technologies such as sensor fusion and AI chips as well. And do contact us if you want to learn more about what we do at TechVision or at Frost & Sullivan. I'd really like to take the time to thank everybody, especially Alex, for joining in on the presentation today. Thank you.